In 1978, a team of scientists was allowed to do something almost nobody gets to do. For 5 days, they had unrestricted access to the Shroud of Turin, the linen cloth that millions believe wrapped the body of Jesus. They photographed it, they measured it, and they pressed strips of sticky tape against its surface to lift off whatever microscopic material was clinging to the fibers. Then those samples went into storage, and there they sat for almost 50 years, waiting for a technology that did not exist yet. This month, that wait ended. Researchers finally sequenced the DNA trapped in those fibers, and the results are now all over the internet, wrapped in headlines about shocked cardinals and secret bloodlines. Almost none of those headlines describe what the study actually found, because the real finding is stranger, and it starts with a vegetable that could not possibly have been there. On this channel we do these cases one way, the evidence and the limits both on the table. So let's do this one properly. First, why this cloth causes more arguments than almost any object on Earth. The shroud is a piece of linen about 4.5 meters long, carrying the faint front and back image of a man who appears to have been tortured and crucified. The image is not painted, it has no brush strokes, and it behaves strangely under photography, which is precisely why it refuses to go away as a question. For believers, it is the burial cloth of Christ. For skeptics, it is a medieval work of extraordinary skill. In 1988, 3 laboratories carbon-dated a corner sample and placed the linen between roughly 1260 and 1390, the Middle Ages. That result convinced a lot of people the case was closed. It did not close. Critics argued the sample came from a patched, repaired corner rather than the original weave, and the fight has continued ever since. So when a new scientific paper appears with the word DNA in the title, the internet reacts instantly, and rarely carefully. Here is what the internet decided this study said: that scientists had recovered the DNA of the man in the cloth, that his genetic profile was 55% Middle Eastern, 38% Indian, and less than 5% European. That this proved the shroud had traveled through the Middle East and India. Some videos went further, that the Vatican was stunned, that a bloodline had been decoded, that the crucifixion itself had been confirmed by genetics. Those numbers are real. They appear in the research. But they do not mean what the headlines say they mean, and the difference between those two statements is the entire reason this video exists. Because the researchers who ran the analysis wrote something in their own paper that almost nobody quoted, and what they wrote points in exactly the opposite direction. The study was published in the journal Scientific Reports by an international team with forensic scientist Noemi Procopio of the University of Lancashire among the authors. They used a method called PCR-free metagenomic sequencing. That sounds technical, so here is the plain version. Older genetic tests worked by picking a target and amplifying it. You decide in advance what you are looking for, then multiply that signal until you can read it. Metagenomics does the opposite. It reads everything. Every scrap of genetic material in the sample, from every organism that ever left a trace, all at once, without choosing a target first. Applied to the 1978 tape samples, it produced something no previous test could: a complete biological inventory of the cloth. Not an answer about one man, a census of everyone and everything that had ever touched this linen. And that distinction is where the story turns. What came back was a crowd. The team detected multiple human mitochondrial lineages, not one genetic profile but many, layered on top of each other. Alongside them, a full microbiome, bacteria of the kind that live on human skin, fungi, molds, and something more unusual, salt-loving microorganisms called halophilic archaea, the sort that thrive in extremely saline environments like the Dead Sea and that do not typically appear in ordinary European storage rooms. Then the plants. Wheat, as you would expect on old linen, carrots, and then maize, bananas, and peanuts. Then the animals, cattle, pigs, chickens, dogs, cats, and finally, Mediterranean red coral. Read that list again because it contains its own answer. This is not the biological signature of a burial. It is the signature of an object that has been handled, stored, carried, and displayed over and over across centuries and continents. Go back to 3 items on that list: maize, bananas, peanuts. Maize is a crop of the Americas, peanuts are South American, bananas are tropical. None of them were being grown or handled anywhere in medieval Europe, because for maize and peanuts, Europe did not know they existed until after 1492. So how does their DNA end up on a cloth kept in Italy and France? The simplest answer is the correct one. It arrived later. Somebody handled this cloth in a room where such foods were present, on hands that had touched them, in a building where their dust circulated, long after the linen itself was made. The same logic applies to the human DNA. Those 55 and 38% figures are not one man's genome. They are the proportions of mitochondrial lineages left behind by everyone who ever touched, folded, repaired, prayed over, or exhibited this cloth. Pilgrims, clergy, restorers, scientists. A relic venerated for centuries collects the DNA of the people who venerate it. The study measured a crowd, and the internet reported it as a person. If that is the kind of distinction you want kept straight, subscribe and stay for the part almost nobody quoted. Now the part that matters most, and the part almost nobody quoted. The authors were explicit. This analysis cannot determine the age of the shroud. In their own assessment, metagenomics is unable to provide robust evidence either for a medieval origin or for a history stretching back 2,000 years. It also cannot establish authenticity, and it was never designed to. Because the cloth has been touched and moved for centuries, they say plainly that isolating any original DNA is not possible. So no, this study did not confirm the crucifixion, and it did not debunk the shroud either. It answered a different question, and answered it better than anyone had before. It reconstructed the biography of an object, the environments it passed through, the hands that held it, the salt and dust and pollen of the places it was kept. Those halophilic archaea are genuinely interesting because they hint at exposure to a highly saline environment somewhere in that journey. But a hint about a journey is not a verdict about a resurrection. And treating the first as the second is how a careful piece of forensic science becomes a false headline within 48 hours. So here is what the DNA really tells us. Not who was wrapped in this cloth, but how many people, over how many centuries, in how many places, believed it mattered enough to touch it. That is a genuinely remarkable thing to be able to read out of a piece of linen, and it required no exaggeration at all. The oldest questions about the shroud remain open, exactly as they were last month. What changed is that we can now trace where the cloth has been, even if we still cannot say when it began. If you want the sharpest version of that distinction, science answered the question it can answer and stayed honest about the one it cannot. Tell me in the comments, which historical object would you most want run through this kind of analysis next? And if this was worth your 10 minutes, leave a like. It is the single clearest signal that careful versions of these stories are worth making. Subscribe, and the next case comes to you. Until then.